Let's diagram some Picornavirus RNA synthesis, shall we? Let's. All right. So first we'll start out with the genome. And so do you remember what's the beginning of the genome? Right, it is a VPG, virus protein genome associated, just like so. And then we're going to have that IRS, just like that. And then blah, 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 a bunch of genes are in here. And somewhere in the middle of the gene, there's going to be the cis acting replication element, or the CRE. So cis here means the same thing as cis and trans in, I think, chemistry. Or in life, right? Cis means same, trans means different. And so if it was trans-acting, it would work on a different, sort of a different strand. Like one strand would activate another, but it's not. It's cis-acting, which means that this particular strand cannot be replicated unless this particular part is there. So it has to be there. That's why it's called a cis-acting element, and it's involved in replication because this is where you make the primers. So a little primer-making factory. And then we're going to go toward the end, and then we have the poly-A tail. A, and that's going to be enough A's for a poly-A tail for today. Yeah. Okay, so step one, our polymerase. And so our polymerase is going to be the 3D polymerase. 3D pol is what I've written up there. And that is the big enzyme that is going to come along and actually do some of this. And the 3D polymerase does not work without its friend, which is VPG. So let's make a VPG in here. Let's color this one green, then we can have a consistent color scheme, shall we? Here we go, VPG. Again, just virus protein genome associated. And so the 3D polymerase is going to bind to VPG. So they are going to interact with each other. So we'll draw a little arrow showing it's going to bind. The VPG is going to bind to this cis-acting replication element, like that. And so if we were to draw this sort of in blown up form, we would have the cis-acting replication element. And I draw it like this because the RNA strands are going to be bound to each other here and just loose up here. And so at the top of this loop, we're going to have a couple of A's. A, something like that. Actually, I think there's only two of them there. There we go. There we go. Okay. VPG is going to sit right here. So there's our VPG. And 3D polymerase is going to sit over the entire thing. So we're just blowing this up to make it a little bit bigger. And so what we do is, since VPG is bound right here, the 3A polymerase pretends as if VPG was actually an RNA. So just like it pretends that RNA is a protein to make the ribosome work, it can also pretend that protein is RNA. So Picornavirus, great at pretending. Yeah, really neat. And so VPG, we're going to add on two nucleotides. We're going to add on a U to match that A, and we're going to add on a second U to match that A. So we're going to turn VPG into VPG with two U's at the end. And so one name for that is VPG, and then a phosphate, and then a U, and then a phosphate, and then a U. Like VPG, PU, PU. And that is the actual primer. And that's its ready-to-use form, VPG with two U's on the end of it. So this thing is going to come down, and it's going to bind. Let's show it down here. It's going to bind to the genome. So we'll draw the tail end of the genome. So da-da-da-da-da, and then A, whatever. VPG with two U's is going to bind, so the U will be here, and the other U will be here, and then those will be attached to, there we go, VPG. And so this is the 5' end, this is the 3' end of the top strand, so this is the 5' end, and this would be the 3' end of the bottom strand. And so then polymerases, like the 3D polymerase, right here, 3D, are just going to copy. And so they're going to copy by adding on extra nucleotides, U, and then they're going to make a copy of the whole thing, boom, until they get to the end of their template, at which point they're going to stop. And so there we go. We have just made a complete negative strand. And so this was the positive stranded genome. And we just made the negative strand copy of the genome. So let's just call it the negative strand. Boop. There we go. That's the idea. And then once we have synthesized a negative strand, so here, let's wipe this out. I've got a little sock for this purpose. Look how professional we are here. So let's wipe all of this stuff off, because it's a video. You can always go back. That's the beautiful thing here. Okay. It's a little tippy. So here's our negative strand. It starts with U. So there's a 5' end. Attached to the end of it, of course, is a VPG, because remember, it's not attached there. It actually starts out the strand. Yeah. And then down to the 3' end. Great. And so now we're going to have our 3D polymerase. So 3D Paul. 3D Paul. There we go. And so once again, it's going to get its little friend, VPG. They're going to get together at their favorite little watering hole, which is the cis-acting replication element on the positive strand. So this is the CRE, Club Cree. Nice. And what's at the top of this? Why, you know what's up there. It's a couple of A's. A and A. You go to the club too long, you got to go to AA. Yeah, why not? I don't know. Stupid, but maybe it works. I don't know if it works. And so then the 3D polymerase is going to just say, well, we got some A's here, we got some G's here, and we're in the club, and everything gets a little fuzzy. Why the heck not? So let's just add a U, and let's add another U to that. And then bam, we've made VPG with 2 U's on it, which is VPG, PU, PU, which is our primer. And that's going to come down here. Now the last couple of nucleotides here that we made happen to be A and A. And so we're going to put our VPG. So let's draw our little VPG on here. There we go. And we're going to have our 2 U's, U and U. They're going to bind to A and A. And then bam, that's a primer bound to a template. And when the 3D polymerase, so 3D Paul, sees that, it's going to say, yeah, I know what to do with this. And it's just going to start adding on to the end of it. This is the 3' end. This is the 5' end of our 2 nucleotide primer. And it's just going to add on to the 3' end, just like so, until it gets all the way to the end. And when it gets to the end, to these U's, it's going to stutter. Stuttering to make the poly A tail. So it always only takes a few U's on the negative strand to make a lot of A's on the positive strand. Polymerases are really good at stuttering. That's just a cheap way to make a poly A tail that seems to come up over and over and over again in not just viruses, but pretty much everything. This is how bacteria do it as well. So there you go. That's the idea. That's how it works. Hope you liked it.